Introduction to macromolecules of life. So during this lecture, I'm going to talk about carbohydrates, lipids, proteins, and nucleic acids. And these really are the four groups of chemicals that are vital to life. So they form the structures. They also act as energy sources, and they perform many processes that are required for life. Let's look at the first one category, which are carbohydrates. And they're made up of the following elements. They're made up of carbon. They're also made up of hydrogen and oxygen. What do they function as? So carbohydrates, they function as an energy source, so either short-term or long-term. So for example, blood glucose in the blood, or sometimes referred to as blood sugar. That's for short-term energy storage. It's used immediately. Glycogen or starch are long-term forms of storage forms. Also, carbohydrates can form structures. So in plants, for instance, carbohydrates will form the cell wall. They form cellulose. If you look at the chemical makeup of carbohydrates, they have two hydrogens for every one oxygen, and that's the same ratio as you see water. So two to one for hydrogen and oxygen, and that's one reason why that's why it's called carbohydrates, is because hydrate refers to water, so it's carbon, and it's hydrogen, it has water in it, or say it's the same ratio of hydrogen and oxygen as water. So if you look at carbohydrates, they're classified according to size. And what we mean by size is we're looking at how many carbons they have. So you can look at broadly in two categories: simple sugars, which are made up of monosaccharides and disaccharides. A monosaccharide is composed of one single sugar. A disaccharide is composed of two individual sugar molecules bonded together. And then you have another group of carbohydrates that are much more complex, and those are the polysaccharides. So we'll start looking at the monosaccharides. So again, monosaccharides are composed of a single sugar molecule, and a monosaccharide is made up of an aldehyde, a carbon chain, and a hydroxyl group. And what it is, I drew a molecule. So this is a sugar molecule. You can see in blue the aldehyde group. You can see red, these are the hydroxyl groups. And then the carbon chain you can see over here. So there's a carbon here, there's a carbon here, and there's a carbon here. And then lines between the atoms, those are indicating a covalent bond. Now, these monomers can be joined together in long chains, or they can form rings or other structures, and that'd be referred to as a polymer if you join several of them together or more. So monosaccharides can be monosaccharides, which we said are simple sugars. They can be classified according to how many carbon atoms they have. If you have a monosaccharide that has 3 carbons in it, it's referred to as a triose. If a monosaccharide has 4 carbons in it, it's referred to as a tetrose. If a monosaccharide is composed of 5 carbons, it's called a pentose. If it has 6 carbons in it, it's referred to as a hexose. So for example, we mentioned already glucose. Glucose has 6 carbons in it. So you can see I numbered them. So there's 1, 2, 3, 4, 5, and 6. So this is referred to as a, if you look on here, pentose is 5, hexose is 6. So this would be a hexose sugar. Okay? And again, glucose is known as a blood sugar. Now, if we bond 2 simple sugars together, we can refer to as a disaccharide. And the bond between them, and sometimes it's called a glycosidic bond, but sometimes referred to as glycosidic linkage. That's the bond that bonds together 2 monosaccharides. So, for instance, if I had two simple sugars, I'm just going to draw it using a ring. Okay, so let's say this is a pentose sugar. So you see there's 5 lines. Where the lines intersect in this diagram, that's referring to a carbon, or that indicates a carbon. So this is a pentose carbon, and pentose sugar. If this is bonded to another, let's say another pentose sugar, right? This would be a disaccharide. Not the best sugar drawing, but that's a disaccharide, right? So example, if you look at maltose, that's formed the bonding of 2 glucose molecules together. Sucrose, which is table sugar, so sugar that we put on foods, that's 1 glucose molecule that's bonded to 1 fructose molecule. Okay, and again, the bond that holds those two, and actually any 2 glucose molecules, any 2 monosaccharides bond together, it's a glycosidic bond that's holding them together. Now, when a glycosidic bond forms, a water molecule is produced. And where does that water molecule come from? Well, hydrogen is removed from one monosaccharide, and an OH or hydroxyl group is removed from the other monosaccharide. And those join together to form a water molecule. Okay? The previous one, this is called the condensation reaction we talked about when we were actually removing a water molecule from the 2 monosaccharides that are bonded together. So in a condensation reaction, that occurs when you have 2 monosaccharides that are bonded together. And actually, a condensation reaction occurs in many different organic compound reactions. It's not just with the formation of disaccharides or polysaccharides. So when you have, or you can describe a glycidic bond as a condensation reaction, or also it can be referred to as a dehydration reaction. They both mean the same thing because you're removing water from it. Contrast, look at, so now when you split apart 2 monosaccharides, you have to put back in the hydrogen that was originally moved from one monosaccharide, and the hydroxide group that was removed from the other one. And this is referred to as a hydrolysis reaction because you actually have to put the hydrogen, oxygen, and its hydrogen and the OH group back in. Right? If we go back with a hydrolysis reaction, you're actually putting water back into it because originally you took the OH and the hydrogen out of those 2 monosaccharides. You need to put them back together in order to break it apart. Now, we also can have a larger group of carbohydrates, the oligosaccharides and the polysaccharides. Now, an oligosaccharide is when you bond together 3 up to 10 monosaccharides. And a polysaccharide, which they can be huge molecules, that's when you bond more than 10 monosaccharides together. So starch is an example. So if you eat potatoes or potato chips, they're loaded with starch. That's a polysaccharide. And it's just a polymer of glucose molecules. Hundreds, maybe thousands of glucose molecules all bonded together. And again, remember, each monosaccharide is bonded to another monosaccharide by a glycosidic bond, also called a glycosidic linkage. Now, polysaccharides, they can act as a long-term energy source. So starch in plants and glycogen in animals. So our liver, for instance, stores glycogen. And we can break down the glycogen when we sleep, and because it's a polymer of glucose molecules, we can break it down to maintain a normal glucose level in our blood. Where plants, have starch, they can use it for long-term energy storage. So an example that we can think of is a potato plant. Potatoes are loaded with starch. Why does it have it in there? Because that's the way the plant stores energy. So it could be used from one season to another. Now, the second category of macromolecules are lipids. So a lipid is made up of carbon, hydrogen, and oxygen. If you remember, those are the same elements that we saw in carbohydrates. In contrast with carbohydrates, lipids store a lot of energy. They store more energy gram for gram than carbohydrates. And as a matter of fact, if you look at the calories per gram of carbohydrates, it's about 4 calories per gram, where lipids is 9. So there's a lot more energy stored in lipids. They function they have a number of functions. One is they form structures in cells. So for instance, you'll find them forming the cell membranes, the plasma membrane itself, which is the outer perimeter of the cell, but also all the membranes inside the cell as well. They act as energy storage, and they take part in cell identity. Also, you see that they're protective. That's another function of lipids. They act as an insulator. So, for instance, you'll notice that some animals live in very cold water, like a seal. They have a lot of fat right under the skin, and that acts as an insulator, which allows them to live in very cold water. So lipids include fats, phospholipids, and steroids. We'll go over each one of those categories. Excuse me. Fats, they made up of 1 glycerol and 3 fatty acids. And what I did here is I drew a glycerol and also 1 fatty acid. So we bond together 3 of these fatty acids to 1 glycerol. You can see there's actually a spot where you can have the fatty acids bond together. Actually, one will bond here, another will bond here, another will bond here. And if you look down the bottom, you can see this here. This is 1 triglyceride, a fat molecule. So you can see in blue the glycerol, which is bonded to the 3 fatty acids. You'll see a lot of carbon-hydrogen bonds. That's where the energy is stored. Those carbon-hydrogen bonds have a lot of energy, very energy rich. Now, when we look at fats, and specifically triglycerides, some are identified as being bad fats, and some are good fats. So the bad fats don't have any double bonds in their fatty acids. They cause you to have the higher levels of bad cholesterol. Unsaturated fats, they have those double bonds. So you'll find the double bonds between the carbons in the fatty acids. That's the good fat. This actually encourages the production of more good cholesterol. So it's good for the heart. Now, triglycerides, fats, and oils. Triglycerides can be fats or oils. What does that mean? Fats are triglycerides that are found at room temperature as a solid, and oils are a triglyceride that are found as a liquid at room temperature. Now, fats are high in saturated fats, which are solid, are high in saturated fats. Oils, which are a liquid at room temperature, they're high in unsaturated fats. So in general, if you're trying to consume fats that are healthier, you want to consume oils. And most of the oils, good oils, are from fish and plants. The fats, the solid triglycerides, which have saturated fats in it, those are generally from animal products. All right, so if you want to consume the good fats, good oils, good triglycerides. You want to consume them that come from plants. Now, second category of lipids are phospholipids. Now, phospholipids are just a little bit different than triglycerides. They have one glycerol just like triglycerides, but they only have two fatty acids. And one of the fatty acids is replaced with a phosphate group. And they form plasma membranes, which is the outer membrane of the cell, as well as the membranes that are found within the cell. Now, if you look at phospholipids, they're described as being amphipathic. What that means is that part of the phospholipid is water soluble, and part of it is not water soluble. So if you look at the triglyceride, I drew a picture down here. Okay, you'll see the phosphate group. This part of it is water soluble, and it's described as being hydrophilic or water loving. Where the fatty acid tails, right here and here, which are bonded to this glycerol, those are water insoluble. They're described as being hydrophobic. And because part of it is water soluble or hydrophobic, and part of it's water insoluble or hydrophobic, I should have said hydrophilic in the first one. So let me try that again. So the phosphate group is water soluble, which is hydrophilic or water loving. And the fatty acid tails are water insoluble, which is hydrophobic. Because of that, that actually influences how they're arranged in membranes of the cell. What you'll see is that the phosphate heads will actually face towards the outside of the membrane, and the fatty acid tails will face towards the inside of the membrane. And you'll see two layers of them. All right, so I'm drawing here. So these are the circles of the phosphate heads, which are hydrophilic or water loving. And the two lines I'm drawing, those are the fatty acid tails, which are hydrophobic. And this whole thing, this is a cell membrane. This would be, all right? So this would continue all the way around the cell. And because of the part of the part of the phospholipid being water soluble and part of being water insoluble, actually cause them to form these two layers. Now, another or the last category of lipids are steroids. And what we need to know from that is that steroids are made up of 4 interlocking chains. We eat them as cholesterol. Your body actually makes cholesterol. Why? Because cholesterol can be modified by glandular tissue to produce some forms of hormones. And now the third category of macromolecules are proteins. Proteins, they're made up of the elements carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. They form structures, and actually, if we look at proteins, proteins have the most diverse functions of any of the macromolecules that we've looked at. They form structures, they transport things in the cell, as well as they're involved in transporting things in the body. They allow movement. Your muscles, they can move because of protein fibers. Antibodies, these are the molecules, proteins of the immune system that will actually attack and neutralize foreign organisms or foreign substances in the body. And then enzymes. Proteins are made up of a number of amino acids bonded together. And the bond that forms between them is called a peptide bond. So proteins are a polymer. Right? We say carbohydrates are polymers. Proteins are also polymers. And proteins, the monomers of a protein, in other words, if you look at the protein, the structure of it, it's made up of individual amino acid molecules all bonded together. So kind of like we saw in sugar, where the monosaccharide was the monomer of a carbohydrate. If you bond together a whole bunch of monosaccharides, you make a polysaccharide. If you look at proteins, if I bond together a bunch of amino acids, then I can form a large chain that forms a polypeptide, which is a protein. If you look at the amino acid, the individual amino acid is made up of 3 parts. An amino group, which is NH2, a carboxyl group, COOH, and then you have an R group, and the R group varies from one amino acid to another. And there's 21 different kinds of amino acids. Your DNA actually encodes for the sequence of amino acids in a protein, because every protein will have a unique sequence of amino acids. So if I have one type of protein, the amino acid sequence will be different than the amino acid sequence in another protein. And again, there's 21 different kinds of amino acids. And we can actually, if I draw a picture to the left, if these little circles, each one of these is an amino acid, you can see how they're I'm making a long chain. This is what happens this is how you form a protein, is you have a long chains, and they can be branched, but long chains of amino acids that are bonded together to form a protein. And again, they're all made up of, and I drew a picture here, they're all made up of the same structure. You have an amino group, a carboxyl group, and then here you have this R group. What the R group just means that that's the part that changes from one amino acid to another. The rest of it's the same, but only the R part changes. And again, there's 21 different kinds of amino acids. So each amino acid, each of the 21 different kinds of amino acids would have the same amino group, the same carboxyl group. They'd have this central carbon with a hydrogen bonded to it, but they would differ in a group bonded to this that is represented by the R group. Now, if you look at proteins, they're organized, or they can be very complex. The shape of a protein determines the function of a protein. If you change the shape of a protein, it no longer has the same function. So we can describe 4 levels of organization of the structure of proteins. The primary structure, that is the sequence of amino acids. And it's the sequence of amino acids that actually will influence the shape that a protein is going to form. So if you change the sequence of amino acids, you change the potential shape of that protein. And that's actually what happens when you see a genetic mutation that occurs. That potentially changes the sequence of amino acids, and that's being produced by the protein that gene encodes for. And that can cause either good or bad mutations. Secondary structure is just the folding, or you can form an alpha helix. Looks like a corkscrew shape of the protein. The tertiary structure is you just take the protein, you fold upon itself. And then in quaternary structure, this is when you have 2 or more amino acids bonded together. Okay, so you can have these long polypeptide chains, 2 or more than that bond together, and they can be very large, and they can form from complications. Now, the final category are nucleic acids. They're made up of the elements carbon, hydrogen, oxygen, nitrogen, and phosphorus. And nucleic acids, they form the DNA and RNA that acts as the genetic material for the cell. Now, if you nucleic acids, there's 2 different kinds of nucleic acids. There's DNA and RNA. And DNA, the chemical formula is 2-deoxyribonucleic acid. RNA is ribonucleic acid. Now, the sugar is what is the difference between the 2 of these. If you look at DNA, the sugar is 2-deoxyribose. The sugar in RNA is ribose, and they are pentose sugar. They're 5-carbon sugar, both of them. Okay? So if you look at the sugar in DNA and the sugar in RNA, they're both a 5-carbon or pentose sugar. The sugar in DNA is 2-deoxyribose, and the sugar in RNA is ribose. Look at the if you look at the now there's 3 kinds of RNA that we'll talk about. Sorry, let me back up. There's 3 parts, and I messed up on this where I said 3 kinds of RNA. There's 3 parts. I should have said on here 3 parts of the RNA. Sorry, three parts to a nucleic acid. Okay, I'm going to erase this because make it more, oh, guess I'm not. All right, so if you look, there's 3 parts to a nucleotide. All right, there's a phosphate group, a nitrogenous base, and a pentose sugar, which we already talked about. But the pentose sugar in DNA is 2-deoxyribose, and the pentose sugar in RNA is a ribose. Now, if you look at the nitrogenous base, then the nitrogenous base is what varies from different RNAs and different DNAs. So if you look at nitrogenous bases, you can find adenine, and the symbol that's used is A. Adenine can be found in DNA and RNA. Guanine can be found in DNA and RNA. Thymine, however, can only be found in DNA. Cytosine is found in both DNA and RNA. Uracil, on the other hand, is only found in RNA. Now, if you look at the types of RNA, there's a transfer RNA, a messenger RNA, and a ribosomal RNA. Now, if you look at the structure of the nucleotide in a little bit more detail, I drew some pictures here of a DNA molecule, and an RNA molecule, so nucleotides. So the blue represents the sugar. So this is our pentose sugar. Okay, so this is the sugar. The red represents the phosphate group. And then the green represents the base. And DNA, we said, has adenine, thymine, guanine, and cytosine. So this base can either be adenine, thymine, guanine, or cytosine, depending on which of the nucleotide it is. The RNA molecule, which I have on the bottom, this is made up also of a pentose sugar, but the pentose sugar is ribose, a phosphate group, and a base. And the base, in this case, with the RNA, is either adenine, uracil, guanine, or cytosine. And then if you look briefly at complementary base pairs, in DNA, adenine and thymine can bond together, and guanine and cytosine can bond together. In RNA, adenine and uracil bond together, and guanine and cytosine bond together. All right, and I'll also include some videos that go into these a little bit more detail that are produced to help understand this. Okay, thank you.